HFSP is, as many of you may not know, is an international program. We are based in Strasbourg in France, in the northeastern corner of France. And it was created back in the late 80s by a decision on the Venice G7 summit of the heads of state in 1987. And in the minutes of this summit, it's clearly written down that these heads of states welcomed the initiative of the Human Frontier Science Program as presented by the government of Japan. And already there, they give us the mission, which is aimed at promoting through international cooperation basic research on biological functions. And all this is what we do. And here you see the members of this program, now 17, that's 16 countries plus the European Commission in representation of all the European countries that are not individually listed here. So we started working in 1990, headquartered in France, and right now we're looking at an annual budget of $60 million, of which about a bit more than 1/3 goes into fellowship support. And really, what is our task? We are supposed to foster and support basic research in the life sciences, and that is very important. It's all about basic research. If you approach HFSP with an idea to develop a new application or develop a new drug, that's not HFSP. So it has to be basic research that's innovative, that extend a frontier. So you try something that hasn't been done before together with your host supervisor. And it is not limited to any fundamental process. All the complex mechanisms in living organisms are open for your research endeavors. We attach very high importance to scientific merit of the proposal. That's what counts. That's what brings you to the next step. It is an international program, and it requires a change in research direction. Carolyn will explain this later. So these three criteria are a very important part of HFSP programs, and they show in the fellowship program in very different ways. This is a map where you can see where our members are located in dark blue. And in light blue are countries together with the members that have hosted HFSP fellows. So it's really a global organization that is available throughout the world. We reach out to many, many applicants from more than 40 to 50 different countries every year. And so it's really a global program, and you're competing in a pool that is stretching over the entire globe with many applicants from different countries. And we were blessed again in 2023 as a follow-up to the initial G7 summit, when in Japan, in Sendai, there was a G7 Science Minister and Technology Summit. And again, the research ministers of the many countries again re-emphasized the value added of HFSP as an international research support program initially established by the G7. And they also commended us for reaching out to support scientists from Ukraine that were displaced by the war. So this is very important to us. And in total, this slide shows you an overview of what we do. On the left side are the funding programs. You see the research grants, the postdoctoral fellowship, and we have an annual prize, which is called the Nakasone Award. Today's webinar, of course, concentrates on the postdoctoral fellowships. We do have this Ukraine initiative, and then we have special HFSP events that would engage our awardees annually at our annual meeting, which takes place in Geneva this July. We have master classes, which are writing classes, hands-on training for writing letters of intent, and we're organizing frontier workshops. So with that overview, just to final comment on my end, if you were to think about other programs and now knowing a little bit about what we are and who we are, what makes us so different. And the reason for that is that we occupy a very unique niche and propose a value proposition for supporting basic research, because we are the only publicly funded international multilateral program where national funders, so the funders of our member countries, put their resources into a single pot without a string attached. So this is really very unique. HFSP programs are always bottom-up, curiosity and idea-driven. It's cross-disciplinary and international. You may propose some research that may entail a risk. We don't punish you for that. To the contrary, I mean, our review committees look for bold, risky ideas. And so feel free to discuss with your future host supervisor something that is, let's say, off the beaten track, you know. You will get a flexible program we are very agile in our administrative operations. And essentially, you are entering into the domain of science without borders, because this is how we understand HFSP. It's an international collaborative, voluntary collaborative effort by nations, and hence lends itself to a science diplomacy approach. If that jumps over, wonderful. So how do we work? So as I said, we have member countries. They provide us with our budget that we in the Secretariat in Strasbourg manage. Of course, we report back to them what we do with it. And in the Secretariat, we run the proposal and award management with currently a staff of 17. There's one central budget, which only responds to scientific merit and excellence. You would be in the position to write annually proposals where there are no priority areas in our programs. It's all across the entire life sciences. It's bottom up, as I mentioned. There are no topical programs every year. It's all open and unlimited in terms of scientific breadth and depth. If you get funded, takes a while until it jumps over. The proposals are then evaluated by an independent peer review committee, which is composed of, let's say, 22 to 24 scientists from around the world, everyone presenting a different background, a different scientific expertise, which makes these committees very powerful and very knowledgeable about the research that is proposed across the entire breadth of life sciences. And then eventually you get funded and you get a 3-year fellowship support that really is unique, because it's not a 1+2 or 2+1. It's 3 years off the bat. So you can do research for 3 years, worry-free. Your salary is guaranteed. Your money is in our bank. And so we guarantee you 3 years of support. And so you can do research as a postdoc, which is probably the best time for you as a researcher. So other than that, I recommend, if you want to find out what kind of research we support, look up our website and you eventually find there what we call the HFSP Science Digest. You can simply Google it, and that gives you a taste of the kind of research in the fellowship program, but also in the research grant, what we support. You get a sense of what it is. If you have questions right now in the Q&A, I think this thing is popping up. Or otherwise, it's always good to write a message to our award office. And the reason why I'm pointing this out is if you write to the office because you have a personal issue or you have a scientific problem that you don't know which project and so on and so forth, it's always good to write, because then you get a written answer back, and that confirms you that you have received an answer to your question and the problem that is maybe solved. And with that, now I'd like to hand it over to my colleague Carolyn to talk you through the details, the nitty-gritty details of the fellowship program. Carolyn, please.
Thanks, Guntram. Thanks so much for an excellent introduction to the organisation. So I'm going to switch speeds a little bit now, and I'm sure you're really all here to find out how to get some funding for your amazing fellowship research proposal. So what I'll do is I'll move into some more specific information about the fellowships program, what we're looking for, what we fund, and then I'll go through a little bit about the timelines and the process, and finally some information on how to develop your letter of intent. And then I'm going to hand over to my lovely colleague Elise, and she'll just walk you through how to use our application platform, Proposal Central. And then we're going to take a very quick look at a couple of successful fellowships, and we'll hear from Samuel as well, which should be really excellent. I'm very much looking forward to that. Just a reminder, we'll take questions at the end of the session. Please do post these in the Q&A box, as Guntram mentioned, not in the chat, because it makes it much easier for us to answer them. And just as we get started, we'll post some links to the guidance for the fellowships applications in the chat. Please do make sure you read through this because it's really comprehensive and also should really cover most of your questions. So first up, I'm going to talk a bit about who these fellowships are for. So HFSP Postdoctoral Fellowships are for early careers who are really adventurous, really keen to move into a different field of research, a new environment, a new country, to learn some new methods and theories and approaches, and who really want to carry out research at the frontier of the life sciences. So HFSP Fellowships come in two flavours. We have long-term fellowships, or LTFs, and these are for people who have a PhD in a biological field who want to really move into a new project in a different field of the life sciences. So maybe moving from genetics to something more microbiology related, or neuroscience out to something maybe more behavioral. Then we also have our cross-disciplinary fellowships, and these are for applicants with a PhD from outside of the life sciences. So something like physics or computer science or maths. You want to move into biological research and really get some experience in the life sciences. So what do we actually support? What do HFSP actually want to fund? As you heard from Guntram, we really want people to take an interdisciplinary approach. We want you to come in with your experience, your understanding from what you've done before, use that experience in a different field, whether that be coming in from physics to biology or moving, as I said, from one field of biology to another. We're really looking for innovative, original projects that can really do something in the future that can have an impact. We're looking for international and preferably intercontinental collaborations. So when you're thinking of moving country, the world is sort of your oyster. We'll go into that a little bit more in a minute. We're looking for scientific research that's at the frontiers of our knowledge, and we're looking for really competitive ideas. So as Guntram said again, high risk, high gain projects are what we're looking for, and we really don't require preliminary data. And of course, scientific excellence is one of the key criteria, but really we're looking for any frontier research topic in the life sciences. So this sounds very bad, but I think we always have to say it. What do we not support? So HFSP tries not to support research projects that could be funded by someone else. So that really applies to applied and translational projects. So if you're looking for a cure for cancer, you probably want to go somewhere else. We don't fund medical, clinical, pharmaceutical research. We don't applied technology or applied engineering projects. We also don't fund studies that are concerned with improving agricultural production and yield. That doesn't mean we don't fund plant science. We really do, but just agricultural production and yield is not in our remit. We don't fund environmental and conservation or agriculture and forestry research. And we don't fund studies that are simply data collection or observational. We want there to be a fundamental biological question that you're going to answer at the end of this project, or try to answer. And we also don't fund research in for-profit environments, although we do allow collaborations with these. And just remember that many projects do still lead to patents and applications for patents, or even translational outcomes. And this is fine, but it shouldn't be the primary aim. These are things that turn out as byproducts for HFSP projects, and that's great. So what does the funding actually cover? So HFSP fellowship funding covers 3 years of fellowships, as Guntram mentioned. These are for people who must be within 3 years of their PhD award date. There are some exceptions for things like maternity leave and sick leave. Applicants from an HFSP member country can go anywhere in the world. Applicants who come from non-member countries must move to a member country. We cover living, research and travel allowances, and we also give a small amount of money for relocation, and in cases where people have children, we also give a child allowance. Do remember that the specific award amounts differ for each country, so really please do check the guidance for this because it's very different. depending on where you go. So what does an HFSP fellowship look like? I think you know that lots and lots of funders will say a fellowship is person, place, project. For HFSP, the project comes first. We want you to propose something that's bold, that's innovative, that's frontier, and that's in the life sciences. And it really should be, I'm going to underline this again, a new area of research for you. But we also really consider the person. We want you to be bringing something unique, something specific, a specific skill, understanding, expertise to the project. We want you to build a new collaboration with a host supervisor that you've not worked with before. But we want it to be really clear that you're the person bringing something exceptional to the project that nobody else could do. And we also want you to move away from your current research focus. And then finally, the place. We want you to move to a new country so that you can develop new skills and resources, and you can build new networks and new collaborations that can set you up for the rest of your research career. So I'm just going to talk quickly through the timeline. So this is our kind of general timeline. I'm sure you've already seen, we open our calls in March, and then there's a deadline for initiation of the fellowships. The deadline for initiation means that you must open a letter of intent before that date in May. And then we have a letter of intent submission deadline. So once you've initiated your LOI, you then have a week to finesse it and complete it, or more than that if you've initiated it earlier, and then you have to submit. Once you've submitted your letter of intent, we then go through some administrative checks in our office. We'll check that you are actually eligible. We'll also check that the fellowship application, the letter of intent, fits to the scope and the remit of our actual call. If you're ineligible, you'll be informed very early, so usually around mid-June. And that also applies to people whose applications may not fit the scope of HFSP. So if you're really applying for something that's applied or very clinical, we'll let you know in mid-June as well. We then send all the letters of intent that are eligible out to our review committee members. So that's 25 to 26 experts from across the world who all have different areas of expertise. So we'll match your fellowship to the people who are best equipped to review and understand this. They will evaluate your fellowship and score them, and then we will take the top 20% of the letters of intent and invite those people to submit a full proposal. And that's usually mid to late August. And we have a final full proposal submission deadline in September. So there really isn't a huge amount of time between when the letter of intent outcome is announced and when you have to submit your full proposal. So just be prepared to possibly set some time aside there. Again, once the full proposals are submitted, we just very quickly run through the eligibility checks again, just to make sure that nothing's changed. And we then send those full proposals out for external peer review by specific experts in the field. And again, these are external experts from across the globe. Once we have those external reviews in, we'll be sharing those full proposals with our review committee members. This usually happens between December and January, and again, they will also evaluate the fellowships. Then they all come together in Strasbourg in person to discuss all of the full proposals that have been received and make a recommendation that then we share with our board of trustees, and they will decide what can be funded. And then the final outcomes are usually announced in March. So I'll change track again now and just talk a little bit about the things you really should do when you're developing your application. So first up is really ensure that the project attempts to answer a fundamental biological question. It's really key that it's not just observational data collection. There should be a fundamental question behind there. Really try and demonstrate that the proposal is creative, that it's frontier. Make sure you're underlining the basic research approach. Think about how it challenges existing paradigms and how it addresses barriers to progress in the field. Make sure you can demonstrate that the project is your own idea and really highlight how this fellowship is a clear departure from your previous research. And also make sure you explain how you will adopt new approaches and methodology. and clearly describe the methods and models that you'll be using. So again, on the negatives, but some things that you really should, common mistakes you should really avoid. So don't propose a project that's just an incremental step forward in the field. We're really looking for those paradigm shifting frontier projects. Don't propose a project that's really closely related to your current research. You really need to be willing to significantly change your field. It's a very odd one, but don't fail to cite key references in the field. Lots of people don't, and missing something that might actually challenge the novelty or the innovativeness of your proposal is quite a common mistake. And then finally, really don't fail to make your unique contribution to the project clear. Really make sure that the reader understands who you are, what you bring to the project that nobody else could bring. So what should a letter of intent look like? These are really short, so make sure it's short, direct, use clear and simple language. Please avoid jargon, people don't like it. Try and hook the reader in with the first sentence, so make sure that your key idea is clear right up there at the front. As I said, avoid acronyms, avoid jargon, avoid unnecessary abbreviations. Try and show the work is interdisciplinary and innovative through the content of your letter of intent. And that's really hard because you have a very small space in which to get this across. Remember, the letter of intent is a really key step in the selection process. So the message needs to be clear, and you need to know that anyone could read it and understand exactly what you want to do and why it's so exciting. And then don't worry too much, because if you do get through to the full proposal, there will be more space for detail. So I'm going to hand over to my lovely colleague, Elise Williams now, and she's going to take you through how to use Proposal Central to apply. So Elise, over to you.
Yep. Thanks, Carolyn. Yeah, so first of all, there are 3 documents that are important for you to access and read. You can find all of these on our website. So that's the application guidelines, the assessment criteria, and the Proposal Central instructions. So just to help you with the website, with the application portal, please read these. This will help you to secure a very successful LOI. This will help you to fit all the eligibility guidelines. And yeah. Slide. Some basic instructions. You'll need to create an account on Proposal Central. I would recommend that you already do this, even if you're just thinking about applying for HFSP. It's good to have already your account set up and just start getting through those steps so you're not doing everything last minute. I have a video of how to do this on the next page, but just so you understand the overall steps, you'll need to go to the grant opportunities page, search for HFSP Fellowships, choose whether you want to apply for an LTF or a CDF. Please remember that you can only apply for one program and you can only submit one LOI each year. And you must initiate your letter of intent before the 5th of May this year. So that means you need to log into Proposal Central and start creating your LOI before the 5th of May, for the submission date of the 12th of May. So here's how it will look when you log in. Go to the Grant Opportunities tab, search for HFSP, choose which fellowship you want to apply for and click Apply Now. Now you'll see the opening page. There's some blue boxes with important information. Please read them and hover over the, let me just pause for a moment. You can hover over the blue question marks for additional information here. Everything that will help you to submit your application will be mentioned here and in our guidelines. To complete the initiation of your proposal, you'll need to type in a title. You can change this later. Please make sure you update it before submitting and then click Save. Now you'll see that the deadline pops up at the top of the screen and you'll have a proposal identifier and LOI ID at the bottom left. Now you can go through and fill out all of the fields that you need for your LOI, research plan, your host supervisor, As you'll see, there's lots of information, so please familiarize yourself with each page. And there's a page for publications. We have quite a few eligibility guidelines about the publication, so please read. So to go back into your proposal, let me go back here. When you're on your home page, just click back onto the LOI ID. It'll take you straight back into your proposal. You do not need to open a second one. You can also go to the Proposals tab. Sorry. Sorry. So here on the Proposals tab, you can, if you've made too many LOIs, you can open as many as you want. I would recommend please just open one and go through with that one. But if you've opened too many, you can also come back and delete the ones that you don't want to use, or click edit to go back in and edit your LOI. You also need to fill out some information on your professional profile on Proposal Central. So this will be your current institution and your degrees. In Proposal Central, you'll see you have a field for baccalaureate degrees and post-baccalaureate degrees. Please put in anything that's pre-PhD in the baccalaureate degree field and anything that's PhD or later in the post-baccalaureate degree. Great. So for submitting your letter of intent in the same application portal, fill out every field, especially the required fields, which are marked with a red star. Then go to the validate page where you'll see, once you click validate, a list of any fields that you've missed. So go through, check these before you submit, ensure that you've filled out everything correctly. Then you can go to the signature page /print LOI. On this page, you can print a PDF of your entire application. Just keep in mind that this is not the exact same PDF that our reviewers will receive. They will receive a tidy, formatted version that contains just the relevant information for the reviewers. The one that you get from here is just a printout of each page that you've filled out. You'll then need to sign the application simply by typing your name and clicking sign. And just confirm that it's been signed, and then go to submit page, click submit. And as you see, because I haven't filled out all the information, I just have a list of fields that haven't been completed. Once you've completed all the fields and click submit, it will go through and you should receive an email confirmation that your application has been submitted. So let us know if you have any problems. You can contact us at fellow@hfsp.org. And any technical problems with the system, you'll need to contact the Proposal Central support line. You'll find that on the website. So I'll hand back over to Carolyn now.
Thanks so much for that, Elise. That was a really great, exciting run through of exactly what we're doing, and it's super useful to know exactly how to use Proposal Central because these things can be really confusing. So before I hand over to Samuel, I'm just going to quickly think about what happens after your HFSP fellowship. So I thought I'd just show you a couple of success stories for some of our fellows. So cross-disciplinary fellowships. We had Alexander Boyes from the United States who undertook a cross-disciplinary fellowship at the University of Cambridge in the United Kingdom, using his engineering background to develop a new bioelectric implant system and surgical technique to just understand if exposing the gut to certain drugs can elicit a neural response. And following that, he's moved on, and now he has a tenure track position at Dartmouth College. So I think we can say that's pretty successful. And then for our long-term fellowships, I thought I'd show you Meita Arruda Carvalho, Brazilian French nationality, who undertook a fellowship in the United States using optogenetics and whole cell patch clamp to dissect the individual contributions of PL and IL-BLA microcircuits, making up fear memory. And she's now an assistant professor at the University of Toronto. So another lovely story of what happens afterwards. So now I'd like to hand over to one of our amazing current fellows. So Dr. Samuel Miravet-Verde, who's from Spain, is 2023 awardee for a long-term fellowship, currently working in ETH Zurich in Switzerland, studying marine microbes. So Samuel, I'll hand over to you.
Okay, I was asking for the permission. So first of all, thank you everyone for the, first of all, nice introduction, but also this very informative session. I remember to be here like 3 years ago now, but what I try with the presentation that I'm about to go over is to just provide a bit on the hints on how I set up a proposal that it was a successful proposal for HFSP. I'm trying to highlight how I combine more or less my former experience with what I wanted to develop in this project. Mainly because I believe one of the most challenging aspects for me to put together an HFSP fellowship it was to understand what was the point between going somewhere else that you didn't in your PhD and going really over those interdisciplinarity flags and changing fields that is expected for all the awardees. So my PhD started with a very apparently a very simple project that is identifying proteins in a genome. Most of you probably, if you work from a biological field, will know that these are apparently very easy and we know how to find them. But the reality is that we have been ignoring for many years everything that is below 100 amino acids in number of proteins. And if you check, you will see that in very well-studied organisms like Escherichia coli, if any, we are just discovering new small proteins and we're really saturated, everything that is longer than 100 amino acids, but not for those shorter. And the reality is that it's very tricky to distinguish these ones from those that are not really encoding in a genome, right? So this was a problem that I tried to address during my PhD. And also to give you a bit of context, this is not something, if it works, yeah, it's not something that is just for the fun of finding proteins. These proteins can really play any role that you see in a regular cell. It's not like they are just concise to a very small function in the cell. They can really play any other type of role. And you can imagine how important this is for the whole functional landscape of a cell, right? But I also want you to focus on the two categories that are on the bottom, that are antimicrobial peptides and signaling peptides, and you will understand now in a bit. So during my PhD, I identified this problem. So I was lucky enough to work in a group that had plenty of experience in Mycoplasma pneumonia. This is an organism model, very small, using systems and synthetic biology. The group was expert in these topics. So I had the opportunity to, as a computational biologist, to play around with different machine learning models back in the days when were not that famous as today. And put together a predictor that I was able to identify small proteins from a genome, but also had the opportunity to integrate different multi-omics strategies from proteomics to genomics, and also setting up new methodologies. And this was all, as you can see, very well focused and narrow to a small organism with a very specific problem in mind, that it was to identify these small proteins that nobody was identifying so far. So when I finished my PhD, I was still considering I thought that this topic was one of those nice ones that can really get you involved or entertain for a whole career. So I started to study what were the lacking points that we were missing, and what else we could learn. And despite the fact that there was some research, as the one that I did in my PhD, for model organisms, and some other studies identifying these small proteins in the gut microbiome, I really wanted to address this globally, like how are these small proteins, how much of the diversity and functionality that we expect life presents are we missing because we are not annotating these small proteins. It was also highly relevant for me to understand how these small proteins evolve. Do they just appear by chance, or are they something that are selected through evolution like any other protein? And as you saw before, that there was antimicrobial and communities or signaling peptides, I really wanted to know what could be the role of these proteins in modulating communities, right? But as you see from my PhD, I was not really working in a group that it was doing this type of research. We were very concise in molecular engineering with a single organism. So I wanted to go over the scales, and that's why I started to get interested in the ocean. So just as a very quick introduction, just throwing some facts, the ocean covers up to 70% of the planet and is in charge of 50% of the oxygen production on Earth. And most importantly, when you start to think about who is doing this and who is occupying this niche, mainly is microbes. They are 2/3 of the biomass of the ocean. And they are relevant not only because of what they do and the functions they perform, they are basically the first link in the food chain. So without them, there's no life in the ocean. They really present an immense diversity ecological niches. This, like if you consider a human as an ecological niche, you would expect a very regular temperature with maybe only changing the diet and a few variables more. But in the ocean, really, you have a wide spectrum of different factors from salinity, temperature to depth. And all these are expected to have an impact in evolution, right? So I was trying to also starting to highlight or to point towards identifying these factors and the role in the evolution of these proteins. Also in connection with the metabolic diversity, really there's bacteria doing almost any kind of biological process that you can imagine in this environment. And the only lacking point at the time that I was asking for this project, it was really that we were at the start of the metagenomics era where we were starting to be able to get genomes. That this is basically what really could help me extending my research to this field. So that's how I ended up finding the group of Shinichi Sunagawa. He's now a professor at ETH Zurich. And you might have heard about his research because he's the lead bioinformatician in the TARA consortium. That is this vessel that goes all over the world just collecting water samples. So Sinis has been in charge of the computational analysis. And maybe for those that don't come from the biological field, it might look like we are very aligned in the expertise, but I can tell you that's not the case. If you work with ecologists, the kind of perspective and scale they work are pretty different to the ones working more on molecular biology, but that's exactly something that I think it was probably recognized giving this project the award, and it was mainly because really going over the scale of my PhD research and trying to scale it up, it was one of the key points of this project. So with that in mind, we propose a project that I will just briefly throw to you what were the different aspects that were written down in the proposal. So first of all, there was an aspect on collecting public data. So we don't go on campaigns. Well, I have colleagues that go on campaigns with the boat and the cruise, but in general, we work with mainly TARA samples and everything else that has been published and is publicly deposited in a website. So we are trying to make the best out of the research that has happened already. And that's how we put together this resource that has more than 12,000 samples. We process and we analyze all these samples. That was as one of the first points on the fellowship. It was really to put together a data set that would be feasible or accessible to study the questions that I was throwing. There was also an aspect in identifying, so in setting up and developing better algorithms to detect these proteins at larger scale. So you can imagine what I was proposing in my PhD was very nice, but it will take years of, and yeah, I think I did the math, so it will be 700 years to really calculate all the small proteins in this type of resource. So there has been also an important factor on developing a better tool for this, also new algorithms that was highlighted during my application. And you have some numbers there. I will not bore you with that, but we are really working at an enormous scale. So we start with 17 billion different small open reading frames that need to be evaluated. We are sitting already in 30 millions that have some evidence of being expressed. And for example, we have 151,000 that could be antimicrobial. So this, for example, tells you already the kind of research that this kind of study that we are running opens for future application. Regarding that, so I just want to highlight that really we are discovering many novel antimicrobials. So this looks like it's a very nice resource to identify, for example, these antimicrobials are basically small proteins that can kill other bacteria. So you can imagine the importance of this in the race of, for example, antimicrobial resistance. But as you see, based on the description of the HFSP, it's not really like this is the main interest of checking for new compounds, etc. There are other bodies that can fund this type of research. But what I was really interested in was how these antimicrobials could just born in a genome based on the conditions. And for example, with the resource that we put together, you can see that temperature is one of the main drivers, and this is something that we are exploring further. But there's an apparent relation with the warming and the higher frequency of finding these antimicrobials in bacteria. So meaning that probably the global warming scenario is also making the bacteria in the ocean a bit more aggressive between each other and killing other bacteria that might be living with them. And this is just one of the type of questions, but it's not limited to just scanning genomes for this. And for example, this is also one of the most interesting questions that I think I can address, and I really don't see this being a good fit for any other agency body, funding body, because as you can imagine, it's a very particular problem with many applications as far as I can see. But something that I really find interesting, that is how many times we can see one of these small open reading frames or proteins being fusioned to a larger one and making the protein a larger version of it or a different version of it. And what we can see is that in this resource we are exploring, there are some archaea in this case, like Thalassarcaceae, that 50% of their genes can be extended, right? And this is something that maybe is not a number that have 1,000 applications, but I find really, really interesting that there's some just life that has higher probabilities to extend their proteins and then have different functionality to the ones that you can see in the same conditions maybe or just in neighboring genomes. With that, I just collected a bit on general comments on what I think you need to take in mind. I think, well, there has been plenty of this in the previous slides. But I think for the letter of intent section, and this is a common aspect, always discuss with your PI. They generally have done this process before. In my case, for example, the HFSP was one of the first time that I was exposing myself as main principal investigator of a project. So always find someone that really has this experience from before. It will help a lot. I would say don't hesitate to apply. I have colleagues or people that have asked me, should I apply for the letter of intent? I'm not really sure. Maybe this is more work for the people in the organization of HFSP, but I really believe HFSP is one of the most respectful in terms of time for the researcher fellowships that you can apply for. Mainly because you don't need to write 10 pages of a project to go over and maybe they get rejected. If the letter of intent is good and is honest and it presents exciting research, very, very high chances that you will be invited to submit a full proposal and then you can really go into details for that. I also keep telling people the conditions is the most tricky, but if you have checked for other applications, you will understand why. And it's really the devil is in the details. Go for everything and check that you really satisfy or check all the marks. Still, in my case, I won't go on details, but HFSP organizers were always, well, the officers were always very, very nice and they helped me and support me a lot during this process. I was already in Zurich when I got awarded, not in the exact time. But you can imagine that it was not exactly moving countries for starting the HFSP, and from a start, I always have all the support, so just go for it. And then for the full proposal, just again, work with the PI. The most important part is combining expertise. I hope it was a good example what I presented. I was not a marine biologist, I was not an ecologist, I was not really working with this topic at all. And nowadays I can consider myself an expert in the topic, so that is what you should seek for. And of course, be ambitious, but keep it feasible. I assume if it's your own idea, you will also understand what are the limitations and what you can do and what not. Go for those, but don't just put everything that is just a fantasy. So always keep some kind of a small thread that you can pull and make sure that that will work. And just with that during the project, I can tell you that it's very, very fun to be an HFSP awardee. There's plenty of yearly opportunities to meet the people in the community. in very nice places. I have the chance to visit very nice countries and different continents. This year, sadly, it will be in my in the country of the research of the grant, but that's still fine. I think it's nice if it's close to home. But yeah, there's one of the few fellowships that really puts together plenty of scientists around the world. It's not really common to find a meeting where you can talk with a medical doctor here, a physicist there, and then a sociologist, and they are doing some kind of crazy model about hyenas in Africa. It's really, really cool. It makes you think out of the box. It makes you also to seek for those aspects of novelty and wow effects in your research, and that's something that I really appreciate. There's still plenty of strong collaboration potential, really. Even me coming from a very niche field in the ocean, blah, 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 I still found people that I'm collaborating nowadays thanks to HFSP. And the most important part is thinking out of the box. I think really in HFSP you will find plenty of people that are really over that edge, right? That they are went somewhere else and a step farther in doing their research, and I think that's very, very, very cool and really extremely exciting and also inspiring for me. And with that, also just to mention and to make some advertisement in there, but there's the Linda O'Larrain meeting that I have the opportunity to participate this year, and this is just a meeting between young researchers and Nobel laureates. So it was me and 500 other students, PhDs, postdocs, and 60 Nobel Prizes for a week. And it was, again, extremely, extremely inspiring. And it's something that also HFSP provided me the opportunity, invited me, and this is also something of the kind of activities that they promote. So just being part of HFSP is really, really useful career-wise and also in extending your networks. So with that said, I just wanted to conclude saying thanks again to HFSP, and you have there some details if you are willing to contact me. But again, you can always do the same with the officers, that they are very, very helpful and kind people. But yeah, if you have any question, I will be happy also to be in touch. So that's all from my side. Thank you very much.
So now I'm going to move on to the bit that you probably all are most excited about. And I'd like to introduce one of our lovely HFSP fellows, Dr. Sandra Reinert, who is German, and she's taken up her PhD at University College London in the UK. And she was awarded in 2024, and she's working on schema representations in the prefrontal cortex. So Sandra, I will hand over to you if you'd like.
Thank you, Carolyn, and thanks to all of you for having me today. I want to start with a little bit of background about myself. I'm a trained biologist, and I did my PhD in neuroscience in Munich. And what I worked on there is our ability to categorize objects, experiences, events in our life. And from work in humans, we've known for a while that when we are in the process of either detecting or forming categories, several brain areas are active. For example, the visual cortex, if we're talking about visual categories, memory areas like the hippocampus and the basal ganglia, but then also associative or higher order cognitive areas like the prefrontal cortex. But we know very little about the underlying circuitry, about how brain cells enable us to form these associations or abstract rules for categories. And so in my work, I aim to address this in mice because we have much better access to individual neurons. We have much better manipulation tools than in humans. And so what I did was I developed a paradigm in which mice were learning new categories and forming rules. And I could show that they can successfully learn and apply those, making the mouse model accessible for this type of paradigm. But then also I developed a methodology on how to record from prefrontal cortex, which is an area that's not that easy to access in mice, especially with methods where we can follow the activity of individual neurons over a long period of time weeks, months in which mice learn a new task. And I could show that neurons in mouse prefrontal cortex represent exactly what the mouse is categorizing. So any learned rule that the mouse is applying. And I could show how this process develops over time, which is something that we couldn't have done in humans before. And so when I finished this work in my PhD, it came to finding a postdoc. And for me, this was really a iterative process of defining a project and finding a host supervisor. And I went through the cycle with several potential candidate supervisors and candidate projects. But what I started out with is asking myself, what do I really care about? What gaps are there in neuroscience that I would like to fill or see filled? And for me, that was the process of abstraction in the brain, which is something that is often seen as quite cognitive, but on the other hand, we have to do it every day and it has such a big impact. And the maybe even bigger driving point is how neural circuits do that basically on a single cell level, because to me, in the end, the brain is largely goo, and goo can make extremely complex computations work somehow, and I want to know how. So after figuring that out, I looked for host supervisors that matched in a way as close as possible to me in what they care about. And for me, the closest match was here at the Sainsbury Wellcome Center as part of UCL, where my host lab cares about understanding how frontal cortex shapes structure representations, which is based on something they have identified before. So I could look at what my host supervisor, the lab, what they would bring to a collaboration like that. And what I saw in them was that they had a very nice characterization on an abstraction that was performed by the brain in an area that I care about. And they were also developing a computational theory on how that could happen in single cells, which is something that I've not spent a lot of time with in my PhD. It was a very systems neuroscience lab, and now my host lab spans the gap between a theory lab and an experimental lab, and that was very attractive to me. And at the same time, my host supervisor was in a situation at the SWC where we have pretty much unprecedented access to tools, to methods that I couldn't have employed before, but that enable me to ask much more precise questions about frontal cortex, which I care about. And then I continued that cycle with, well, what can I bring to that project? What makes the connection between me as a postdoc in that lab unique? Is there anything that I can put forward that isn't possible in any other combination? And so in defining that project, I tried to bridge that gap by highlighting the methods that I've developed that no one or very few people in the world have been able to employ, which is imaging, which is a type of neural recording in frontal cortex with very extended long-term tracking of the same neurons, and at the same time, extensive understanding on how to build behavioral interrogation methods and tasks for animals that can answer questions in a way similar to, like, that can pull cognitive behaviors out of animals in a way that can be compared to what humans do. And so together, my host lab and I developed this project in several cycles of working out how they can best support my trajectory in working from what I knew based on my PhD work and where I want to go when I hopefully apply for independent funding at some point. and how I can best contribute to the lab by providing expertise on the methods and bringing, a new technology to the building. So that was, and then pretty much these were the main points of focus that I then tried to highlight in my letter of intent. And that's pretty much it.
Thanks so much, Sandra. It's a really lovely description, actually, of exactly what we're looking for, like how you thought about what you could get from a host supervisor, but also what you could bring. So that kind of unique approach from you, but also what you can stand to gain and learn. So thank you. That was absolutely excellent. Really grateful to have you join us. So like we said before, we're coming now to the question and answer session. Please do put any questions that you might have in the Q&A box, and we'll try and answer as many as possible. If we don't get to your question, or if it's really super complicated, then please do just pop us an email afterwards. Like Sam said and Guntram said, we're here to help. So just email us and we will try and answer you. We've popped up the links for emails, for the website and for socials just on the side here. But now I'm going to escape from my screen share and hopefully we can start looking at some questions. Cool. Thanks, Guntram. Thanks so much for being here with us.
Nice.
So I guess we'll start. So first question up is, how different should the field from our PhD or previous field of research be? Does it matter if the proposal is in a similar field? I mean, I think, as you can tell from what Sam was saying, you really do want to be thinking to move as far away as possible from your original research, but try and make sure that you're still bringing that unique contribution. Your expertise is super valuable. It's important that you have something to contribute. And sometimes the way you contribute might be really different to what you're thinking about, but it does need to really move field. How many postdocs are funded out of the top 20%? So we basically normally have around a 7% total success rate from letter of intent to awards. And then we have 45 to 50 from the full proposals, but that really depends year to year and budget. A simple data collection study is not eligible. So that would mean literally if you went out into the field and decided that you were going to, let's say, analyze the genome or analyze, let's say, collect the genomes of loads of microbes in a particular soil sample. And that's all you're going to do. You're just going to collect the data, say, that's it, we have the data. What we're asking you to do is maybe say something like, in a particular area, are these microbes expressing similar antimicrobial genomes, or in these particular T cells, how are they responding to a particular stimulus? We want a fundamental question under there. So everyone has to collect data. It's part of doing research. There is a point at which you will need to collect data, but that can't be the only thing that you're doing. You have to go further. You have to use that data for something. So new or different. So I love this question because actually I've read the research funded in recent years and it all seems so extraordinary and groundbreaking, and almost all of it is laboratory based. And you're in the field of nursing. So I think the issue, what you need to think about there is, if you're in the field of nursing, what you don't want to do is then move and undertake a clinical study, because that would absolutely have to go to a different funder. What you'd want to do is think about asking that, again, go back to that kind of fundamental question, whether it be neurobiology, whether it be immunology, anything like that. Think about that basic biology question how you would ask that, so what you're setting out to answer. You don't want to do something totally clinical. And as we've mentioned before, data collection, you will have to collect some data. That's normal, but that data needs to be collected in relation to the question. And if your supervisor is from a particular university, does it mean you'll be working for that university? Yes, normally. People do sometimes have two supervisors, so you have a primary host supervisor and a secondary host supervisor. You'd normally find yourself working most of the time at the university where your primary host supervisor is, and they will be the people who will pay you. But sometimes if you have a secondary host supervisor, if they're in a different institution, you might find yourself going and spending some time there, either to learn techniques or carry out a particular experiment or understand how to do a particular bit of data analysis. I hope that answers your question. So if a PhD in chemistry and chemical biology and some background in biochemistry, I always think, so think carefully about how, so this is whether you're a PhD in chemistry and chemical biology with some biological background fits to the cross-disciplinary fellowship, especially if you're focusing on understanding innate immunity. This requires new techniques. You need to think about how to justify whether you're a cross-disciplinary fellow or not. If you go and emphasize, oh yes, I've done lots and lots of biology, then you probably want to think about an LTF. If you feel really that all of your expertise is really focused in chemistry, biochemistry, really further onto that side, then it could be a CDF. But it doesn't matter. If you apply for the wrong one, we will reassign you to the right one. So if you do kind of fall into that gray area where we're not completely sure as to whether you're an LTF or a CDF, we can ask our review committee as well if they will change it. So it doesn't affect how you're assessed. CDFs and LTFs are assessed in the same way. They don't have a separate budget. So if you do get it wrong, it is not an enormous drama. Don't worry. So I find it difficult to know how much detail to write in the LOI about the experimental model. So if I use a mouse model, is it okay to describe how it works for my aim in one sentence, or do I really need to describe it? Think about how much space you have, how important the model is versus how important the actual question that you're asking is, the actual hypothesis is, and try and balance those. You do need to make sure that it's clear which mouse model you're using, but you don't need to describe every in and out of it because you've got 3 references. You can use one of those as a link to describe for the full description of the model. That's okay. So how different should this shift in research be, and can the model organism be the same, or does it need to be different? How likely is a different question using the same model organism but somewhat related to the PhD to be funded? I can't tell you how likely anything is to be funded because not only is it really difficult to predict, but also our review committee changes slightly year to year. So we have new members coming in every year. So the way that they look at things changes every year. We do get people working on similar model organisms, but if it's closely related to your PhD, you probably want to think about a different funder. If what you're bringing to the project is the experience with that model organism and you're going to a completely different field and looking at something completely different in it, then that could be fine and could be interesting. But I really can't tell you how likely you would to be funded. That is a question that doesn't really just go on the topic. Whether you're likely to be funded goes on how well you explain your letter of intent, how well you write your research proposal, how well you put everything forward as well. What was the success rate of the last call? Was about 7% for the letters of intent, and that's kind of where we are. So my case seems to be between the LTF and the CDF, computational background, but always work directly in the life sciences and intending to propose a project in a different life science subfield. Could you confirm that an LTF is appropriate? I would suggest that you apply for an LTF, and as we mentioned, if we or our review committee feel that you would be better suited to be a CDF, we can just change that over for you. If the number of CDF applicants increases, will the funding rate remain the same as in previous years? We rank, so the CDFs and LTFs don't have a separate budget, but they all score in the same way. So basically, if we got, if there were more CDF applications that scored really highly, then we would be funding more CDFs. I hope that makes sense. If my proposed project is innovative and in a new research field, would the proposal be rejected if I wouldn't move country for the proposed research? Yes, you have to move country, unless you've not lived or worked there for 12 months. So if you were, let's say, if you've done your PhD in the United States and you applied to do a fellowship in the United States, we would unfortunately have to contact you and say that you were not eligible to apply. If you'd done 6 months of research in Canada and then left and gone back to the United States and then applied to do a fellowship in Canada, then you would still be eligible. And if you've got really detailed questions about eligibility, please do just pop us an email. That's what we're for. So yeah, I guess the answer to how important is moving country for the application, it's critical. How much should the project differ from the host's previous projects? You want to, for this, you really want to make sure that you're showing what you're bringing that's different. So you need to show in the project that, yes, sure, you're leveraging some of the expertise, the understanding, the knowledge of the host, some of what they do, because otherwise there's no point going there. If you're not using some of what they do and some of their knowledge, it's not helping you. But you should still be able to really clearly demonstrate why it should be you and not just any other postdoc in that host lab that can carry out that project. I hope that helps. May the host supervisor be from a mathematics background if the research is applied to biological problems, or is prior experience in the life sciences necessary? Again, so you need to show that the supervisor is the appropriate person to supervise that research. If you're coming in with really strong biological background and using mathematical models, and you have a maths supervisor who is absolutely excellent at that, that's great. But remember that the focus for HFSP really is fundamental frontier life science research. So you may want to consider something like having a second host supervisor who is an expert in that biological field to help you out. In the case of a project that uses a life science question using machine learning approaches, is it acceptable for the primary host supervisor? Yeah, to be computer science or engineering, as long as the core research question is rooted in the life sciences? Yes, as we said, absolutely. But do think about whether there's enough expertise on the team to help you to carry out your project. You want to know that the right people are there behind you to help you to be successful. I have one common question regarding the new or different field or topic. Can I get a clearer view of how different the field can be? For instance, can I switch from systematics and taxonomy field to ecology? They're very different. There's the common regions. So how wide does the common region need to be? It really depends, actually. So this depends hugely on which question you're asking, how far you're moving, and where your expertise is. And that honestly is something that can be really difficult for us to judge. And it's something we really ask our review committee members to think about, because they really understand. I mean, I'm an immunologist, so if I turned around and said, I don't know how different that is, that might be just me looking from an immunology point of view. So you need to think about that. You need to think about how you can argue in your letter of intent and in your full proposal how different the field is, because it's down to you to show that you're making that change. It's down to you to explain it and make it clear. Hopefully that works. If I wish to stay in the cancer domain, what are my chances for the proposal to be accepted? And if I switch to a different domain, for example, again, we cannot, there's no way we could ever predict what your chance of funding would be. It's really difficult, and it's not that I don't want to say, it really is, you cannot tell from what comes in, what's going to be funded. If you stayed in the same field that you're working in now, you might find that at the letter of intent stage, the review committee say you're not making a big enough change in research direction. So you might find that your letter of intent was rejected. But it could be, you might be just using cancer as a model and moving to a completely different field, in which case it might be different. So again, it's up to you to think about how you put forward the change in research direction, the change in field, and how you argue that case. So I have a question about funding medical research near life science HFSP proposals. So my proposal would focus on uncovering new regulatory immune circuits in a specific disease to validate the identified circuits and using a platform currently developed as a potential therapeutic. So would this be too close to the funding of therapeutic approaches? Again, this is up to you to argue how different it is and why it's not therapeutic. So you need to make it really clear in that letter of intent what the fundamental biological question is, why you're using something that could be a therapeutic, but you're using it to answer a fundamental biological question. So it's really about how you make the case for your funding to fit to our scope. It's about thinking about, that's the scope, this is how I want to do it, how does it fit to that? So I saw one of the answers, you receive a much larger number of LTFs compared to CDF proposals. Do you have the number of or an approximate of LTF to CDF submissions or an approximate ratio? And are the acceptance rates similar for both fellowship types? We don't really, we usually receive, let's say, about 12% CDFs compared to 88% LTFs. So we have a lot more applicants coming in from a biology background. The acceptance rates are, so for LOIs going through to full proposals, are probably similar, yes. And again, remember, if you do get it wrong, it's not a drama. We can change that for you. So it's not as terrible as it sounds trying to work out which one you are. How do reviewers evaluate the balance between proposing a strong shift from the PhD field and demonstrating sufficient expertise to carry out the project? You'll find weight these things very evenly, because it is very important that you have the expertise and that you're exposing yourself to learning new techniques. And that's why when we're also talking about the host supervisor, that's why you need to think carefully about the host environment to make sure that that has all of the critical things that you need to carry out that project. So not only do they have some of the expertise that you're missing, but they'll be able to support you to learn those new techniques, to learn how to use a new model organism or to culture different cells or work with organoids. And that's why you also need to think, if you don't have the expertise, all of the expertise that you need, that potentially a second host supervisor might be a really useful thing just to help you with something where you feel like you don't quite have that expertise on board. And then, is it important to change the experimental system or model organism, or is a conceptual shift in the research question sufficient? This is down to you to argue your position, to make it clear why it's a change. And if you feel you could use a model organism that you've worked with before, but to a completely different field, why not try it? But make sure you make it clear what the change is and why. I'm a systems biologist working on mapping the landscape and further understanding of the dynamics of cancer development and progression using network and mathematical modeling-based approaches. Again, is this expertise considered translational? It's up to you. You can use your expertise to do a project that's not translational. So it's not the expertise that we consider translational, it's the underlying question. You know, you can't come in with an underlying question that is, how do we cure this, or how do we develop this therapeutic, or how do we develop this diagnostic tool? But you can go in and say, how can we understand more about how these cancer cells work? It could be a more fundamental question. Okay, Liliana, how are we doing with the questions? We've got a couple more. So, how related should the host lab be to the submitted proposal? I think we've really already answered that, but just remember they need to have the expertise to support you, but you need to bring something really different that is going to really help the host lab to answer that question, to fulfill that project in a way that they couldn't without you. All the LOD projects illustrated seem to be largely empirical and experimental in scope. Would a project mostly oriented towards theory and mathematical modeling be suitable for a proposal, provided it attacks a clear research question based on empirical observation? As long as you're not just carrying out an empirical observational project and you have a really clear research question, you can use the techniques that are appropriate within that field of biological science. So hopefully that helps. If I tried to switch from plant ecology to marine ecology to apply a more quantitative approach to a field that's mostly observation-based, is that enough of a field switch? I mean, plant ecology to marine ecology is fairly different, but still ecological. So again, you need to make sure that you're explaining why it's different, what your expertise brings to the project, and where you're going to go from there. Does it matter if you use a model system commonly used and developed in the host lab, but it's something you as an applicant have never worked with? If you've never worked with it, that's different for you. But again, you need to show what you're bringing that the host lab couldn't do without you. So how are you going to use that model system in a way that complements your expertise, your understanding? Where are you going to take it? Are you going to study a topic that the host lab had never studied before? Are you going to use the model organism differently? What are you going to do that's completely outside of the field that the host lab is doing? And that doesn't mean it can't be related, but it does mean you need to show that unique contribution that you have. If someone is to step outside of their discipline research area, how does one convince HFSP that they can carry out the proposed research? Do you know, I might actually ask Sam to answer that one, because that's really hard. And I think, Sam, you've probably got the best answer here.
Sorry, I have some dogs barking in my place. So can you repeat the question very quickly and I go for it?
Yeah, absolutely. So the question was, if someone is to step outside their discipline and research area, how do you convince HFSP that they can carry out the proposed research?
Yeah, I think the easiest there is don't really put yourself in the box of I need to become an expert in something different. It's about what I know, what I can bring to this other field, right? So from my perspective, I still really believe and I still work as a computational biologist overall, but the field of expertise is extremely different. So I still can claim that I can use my skills in statistics or computational modeling, but where I apply these technologies or methodologies are pretty different. And then in the end is what we do every day, right? Looking for bibliography, finding the way to do things, and trying to convince that you know how to implement those in a new field. But don't stress. I would say my recommendation, I know it's stressful, but don't try to look and becoming an expert in something else. It's more about finding new ways and new possibilities within the scope that you are an expert. So,
thank you, Sam. I think that was the perfect answer. So, is it necessary for either the applicant or the host institute to be proficient in all the techniques required for a proposed project? I'm going to skip the specifics there and say no, but the combination of the applicant and the host should be able to cover that expertise. And again, if you don't, then you need to show that you've got support from someone else who will help you with those extra bits. I hope that helps. If you've got a really complicated question, please do just pop us a message and we can help with that. And then I've worked on T cells in metabolic disorders during my PhD. Can my proposal be focused on infection immunology, such as host-pathogen interaction in mouse models? Again, that's a very specific, it is a very specific question. It is quite different. But you need to make sure, remembering that our review committee members are not all immunologists, that you actually make sure that you describe exactly why that's such a big move, and whether you're going to focus more on the actual infection or infective microbes, or whether you're going to focus on different immune cells other than T cells. So you really will need to show how that's different and how you're going to use new techniques, how that's going to stretch your knowledge, how it's going to expose you to something else. Questions? So first one up is, can you define or give some examples of what counts as applied technology or engineering? So I think probably one of the easiest examples that I could give is something interesting we had, where somebody, if you would like to build a piece of equipment, like build a flow cytometer, and that's what you want to do. You just want to build a new kind of flow cytometer that's maybe going to pick up on cells slightly differently or will read in a different way. That's really exciting. That's absolutely amazing, but that's just developing the technology. If you wanted to build a flow cytometer to answer a specific fundamental biological question, so you wanted to build it because there is no way of visualizing how a particular cell is reacting with the technology that's available now, then that is when you would be actually doing something more than just building something or making something. I hope that helps. So we want to ask a question regarding the clinical focus that's excluded from HFSP projects. Would a project that involves clinical populations, for example, depression, but aims at trying to improve our understanding of biological mechanisms in this group, be acceptable? So the answer is, this is where you need to make the argument that what you're not trying to do is develop some kind of therapeutic or some kind of cure. What you are trying to do is really understand what's going on in this particular cohort. And that is really up to you in your letter of intent, and if you're lucky, your full proposal, to really make that case for what the biological question is, what you're going to answer, and how that is really basic science that you're doing. So what defines the degree of new area? For example, is this expected to be moving between two completely different fields, for example, molecular biology to physics or optics, or will shifts within a subfield of one's current discipline be considered equivalently? For example, researching a different model organism such as rats and humans. So for that, I would really say, again, this is down to you to make the case as to what's different. So what we're not expecting you to do is going, I am doing visual neuroscience in mice and I'm looking at this specific neuron and I'm going to look at exactly that in rats. I don't think anyone would consider that to be a big enough move. But if you're going to be saying, I have great experience working with zebrafish, and I'm going to go and take my experience working with zebrafish into a lab that doesn't have zebrafish experience, and I'm going to use some of their understanding, their knowledge, but bring my methodology, my techniques with me and learn something new. That's what kind of a shift we're looking for. But again, it is really down to you to make sure in the LOI you explain what's different, what you're bringing, what's new for you. So we're talking about basic research. If a pathology is framed as a deviation of normality to research an underlying cognitive mechanism, does that work? You can research disease conditions like that. That's fine, as long as, again, you're asking a basic question. There needs to be a fundamental biological question underneath. You can't say, I want to study this disease because I want to find a cure. We're looking at you saying, I want to study this disease or this condition because I want to understand something about it. Hopefully that's vaguely helpful. If my expertise and knowledge from both my current research and PhD work are not well aligned, it becomes very difficult to convince a host lab to support my application, as it may appear that I'm shifting between unrelated areas within the biological sciences. I think that's quite a complex one if you're saying you maybe move from BSc or master's level to do something different at the PhD. And I think you really need to work on finding a supervisor, communicating with a supervisor to make sure that they understand where you come from, how excellent you are, and where you want to move to, and make sure that that trajectory makes sense and that you can explain it. Because if a supervisor can't understand your trajectory, it's very likely that our review committee members will also struggle to see that trajectory. There are opportunities for dentistry or oral health related projects. If it's basic biology, then yes. So if you're looking, if you're trying to understand something basic about teeth, I guess, or gums, or any kind of something to do with that, then yes. But if you were trying to develop a new composite for fillings, then no. It can be hard to demonstrate that the project is genuinely yours when you're changing topics and moving to a lab that's an expert in the new area. So how do applicants usually demonstrate that the project's usually theirs? I'm going to ask Sandra to answer this one because I think this is a really good one for her.
Yeah, I think this is definitely what I was thinking about. And this is a difficult bridge to gap between you're trying to move away, you're trying to grow from where your PhD was, but at the same time, you're trying to show that your contribution is valuable. So for me, the way I did that was to bring a method. And I'm aware that maybe not everyone has that it doesn't look identical for everyone that applies. But for me, my host lab is more experienced in this very specific question or the topic that we're asking. But I'm more experienced in the method that I'm using to interrogate the system. And so for me, the growth is conceptually, and for the lab, the growth is in access to new tools. And that worked extremely well for both of us, but I'm sure it can work the other way around, or it can be two different topics that get combined, two different experiences, something along those lines.
No, that sounds I think that's exactly right. Thank you, Sandra. That was excellent. Can I collaborate with supervisors who were co-authors in my previous research and not my main supervisor? And the proposal itself is not related to my PhD or in my previous research. I'm afraid the answer is no. You should not have co-authors from your previous research as your new supervisor. So there are some exceptions, but really for things like consortium papers. So I would strongly recommend that you actually just email us to ask that specific question rather than try and figure it out on your own. We're here to help. For the cross-disciplinary fellowships, how important is it to propose a project that incorporates my non-life science doctoral research area? I'd like to combine both disciplines in my future independent career, but ideally I'd like to focus my research and training on the life science discipline in my postdoc. That's absolutely okay. It's fine to try and focus on the life sciences, but it is important to show why you're the right person to do that project. And it is, you will have skills that your host lab want to leverage. It's really important that you show those, because otherwise, if you're just going to do a project in a host lab that any postdoc could do, that's not necessarily what the review committee want to fund. They really want to fund something that is going to be incorporating you, bringing in that part of you, your experience or your expertise, or even something you're just really interested in and learned lots about. That's what they want to see coming in, not just you as any old postdoc in a lab. Is working on a completely different scientific topic, but with the same technique newly developed during the PhD, considered as changing area? It's up to you to argue this. It's definitely possible. People do bring technologies. I think, as Sandra just said, she brought methods. So, and it's good, because then you'd be showing that you're bringing some expertise, but it really is down to you to make the case as to why this is different and why you're changing fields. How are the reviews of the LOI and full proposal? LOIs and full proposals are both reviewed by our review committee, which is about 25 to 26 members, all with different expertise, and they're from across the globe. But at the full proposal stage, we also send the full proposals out to international experts in the field. So we really try and make sure that we have specific reviewers, have really specific expertise that's tailored to those proposals as well. And our review committee members can use those reviews to help inform their decisions. What level of overlap between a PhD and a postdoc project is allowed? For example, if the overlap is only general, so during the PhD, many projects on comparative genomics of phages and bacterial immunity, and a postdoc project on phage-host interactions and how phages affect translation, would that count as too similar? So would staying within microbiology and within virus-host interactions be considered a problem, or given that the field is huge, is fine? Lots of the fields are huge, and it very much depends on what the review committee members think. So, as I think I keep underlining, it really is up to you to really demonstrate that it's a change for you. So you need to show a difference. You really don't want to say, well, actually, I've been working on bacteriophage now, and I'm moving on to work on bacteriophage again, and this is the extension of my PhD work. That's not fine. You need to show how it changes, what's different between your PhD and this project. So if you feel like it's a really big difference, then that is up to you to make that argument, make that case, and make it clear. So I would say with a PhD so my PhD is in neuro-oncology. Will I still be eligible if I'd like to apply for a proposal in neuroengineering or neurodegeneration? Again, you have to make the case as to why it's different, what you're bringing and what you're going to gain. If the research topic and question is the same, but the approach is different, is it regarded as changing field or topic? I'm interested in the effect of early life stress on brain development and switching from human brain mapping to mouse brain imaging. I mean, again, I think, Sandra, you look like you want to say yes, so go.
I mean, yeah, I don't think I could add anything beyond what you were saying, that this is like up to highlighting what's different, right?
Yeah.
To me, it does sound very like slightly like you can ask fundamentally different questions with those two methods, and if that's how you manage to highlight it, then
yeah.
But I don't know.
Yeah, I mean, there's lots of things you can do with mice that you can't do with humans. So it's again, yeah, thanks, Sandra. It's up to you to really make that case, make that argument. How does the LOI committee assess if the HFSP committee assess if the LOI significantly differ from the applicant's PhD work? Yeah, so Sandra actually changed quite dramatically, I would say. And I think what you need to remember is that that when you submit your LOI, you do have to submit some details about your PhD project. And those review committee members, they see a lot of these. They do see, if you think we get lots of applications, there are only 25, 26 members, they see a lot of LOIs. So they do get very good at being able to say, yes, this is very similar, or no, this is very different. Also remember that we do make sure that we assign your LOIs to review committee members who have either they've got the methodology expertise or they've got the subject matter expertise, or a combination of both. So it is going to an expert, they will know. I have a PhD in veterinary medicine and I've experience working in a dry lab doing data-driven research. Can I prepare a research proposal that's data-based and does not include any experiments? So still staying in data-driven mathematical modeling research. Again, I mean, the mathematical modeling data science field is really huge and continuously growing. So it does depend what you're doing, because it's good to bring a technique, but remember that you should try and expose yourself to new knowledge and new approaches as well. So it is down to you to try and think, what am I getting out of this? You know, is this just moving on with my PhD, or am I going to get something? Am I going to learn something new? This is definitely for you, Sandra. So I wonder how Sandra found a lab that she was willing to help her in shaping the proposal when pulling from research groups with no previous collaboration, pulling from research groups with no previous collaboration, because not everyone's happy to help. So they want to know, what are the green flags to look for? What are reasonable ways of identifying and approaching target labs, because they've been struggling with this. Thanks, Sandra.
Yeah, of course. I mean, this kept me busy for a while. I was like, after finishing my PhD, I spent nearly a year developing both my own vision on what I want to work on, but also reaching out to target labs. And in a way, for me, the screening process was based on papers that I've read previously, people that have impressed me by giving talks, or research, like leaders in the type of research that I care about, but also evaluating where they currently are. That was a big factor for me. And after that, I made a big list of candidate labs, and I just cold emailed them. And I try to be as clear as possible about who I am, where I'm coming from, and what my goals are and what my interests are. And in the labs that got back to me, we set up Zoom calls, we set up chats, sometimes more than one, before we sort of had a feeling for, will this work, will this not work? And I did go through that process with a few labs. So not just I just selected this one lab and everything worked out. But for me, what were massive green flags were in this one-on-one meeting with a potential supervisor. Do they listen? As in, am I able to provide a contribution to a vision? Am I able to steer the project that I would be working on for the next few years? And in my experience, there were a few PIs that I chatted with who had a very specific project in mind that they wanted a person to work on, and my contribution wasn't really looked for. And that were labs that I just decided not to go for. Another one that I find extremely useful was, like, I was always clear from the beginning that I don't want to just develop a project and then work on it funded by the host lab. I was always quite clear from the beginning that I do want to use a project like this to apply for fellowships, because it's but I mean, you're here, you know about it, how crucial it is for a later on career for getting funded, to get postdoc fellowships early if you can. So somebody who was receptive to me spending significant amounts of time on applying for fellowships was important to me. So I think those two.
Perfect. That's a really wonderful answer, actually. I think it's excellent. Would a project with a component in the philosophy of the life sciences be eligible for the application, either as a major or a minor supporting component? Do you know, I honestly would say we support basic science, but we also want to support our fellows to do things around that. So to do outreach, to do public communications, to communicate their science. So I think as a minor, then yes, but obviously a major, philosophy of life sciences really isn't a basic science. So you're that's a background thing. And I think what I would say is that's a really interesting thing to add in, but it shouldn't be the fundamental underpinning part of your research. So would the other way around up to the Sandra case be of interest? I'm bringing a topic closely related to my PhD in completely different models, so moving from invertebrates to mammals. I think if you have a look at the fellowships awards on our website, you will see that there are a couple like this. So again, it's about the case that you make as to how it's different. And obviously it's quite a big shift from invertebrates to mammals, but you really need to make it clear, that you're moving from, I don't know, Drosophila to studying whales. And that was quite different in size. But you're really moving and you're studying something, but you're kind of bring expertise in that. So just have a look at the ones that are on our website, because you actually might find some interesting examples in there. Does HFSP provide mentoring or career support and training after the fellowship starts? Some fellowships assign mentors to fellows. Does HFSP have an equivalent? At the moment, we don't. So what we do have is our awardees meetings. I think you'd be surprised at how interdisciplinary, different, entertaining these are. It's a really good chance to meet peers and meet other people in similar situations, but also some of our grant awardees, some of whom are people who've had fellowships in the past and moved on from there. So we don't have a mentoring scheme, but that doesn't mean that you can't also pop a mentor into your fellowship application. So there's nothing standing between you and looking for a mentee yourself. But it's also a really good idea, so thank you. Could you elaborate a bit more on what your definition of high risk for a project is and how much is desirable? Should we include contingency plans in your LOIs and proposals? Yeah, bizarrely, risk mitigation is important. So it's good for a project to be risky, but it also, feasibility is actually quite important. So you do need to just demonstrate some degree of feasibility. And if it's something that's incredibly risky, then a bit of, then some sensible mitigation plans are there, especially if you've got something that's maybe you've got aim one, you're wanting to collect data from something and you're not sure if that actual method of data collection is possible. And then if aim 2 and aim 3 depend on it, you really do need to think about what the plan would be if aim 1 goes pear-shaped. Can you elaborate on non-material support of a Human Frontiers Fellowship? Is it beneficial for future Human Frontier grants? Is there an alumni network? So the non-material support really does come down to that kind of awardees meeting. There's, I've met a lot of people who go, oh, those HFSP fellows, they're all amazing. So I think that's a nice thing to turn around and see. That's really fun. But it's also that there are chances to do other things. And the lovely Liliana has a good answer for this because she's our comms officer. So Liliana, over to you.
I just want to add that we also have a Slack channel for fellows. So it's a place where they can exchange opinions, opening calls for participating in meetings, conferences, and even job offers. So it's quite lively there, and it's thought just for fellows, specifically for fellows. So it comes also with your fellowship if you are selected.
Thanks, Liliana. That's a great answer and super helpful. So yeah, there are other advantages. It's not going to help you get a grant, but having been successfully awarded a fellowship kind of shows that you know how to write an application, how you know how to write a grant, and that's always helpful. So having had funding makes you look like a good bet for future funding, if that helps. So it certainly makes it easier for you to write a grant in the future. The LOI has relatively limited space. Does the part where we try and make a point about how far or related our PhD work to our proposed project is go in the summary, or in the section where we discuss our PhD project? It needs to go into you need to make the LOI clear that it's a departure. You don't need to discuss it in depth. It's not something you need to write paragraphs and paragraphs about. Just need to make it very clear that, you're moving away, that this is what's new and this is what you bring. Sorry, that makes it really hard because there's not a lot of space, but you do need to be able to say, this is what I'm doing. This is what's new to me. This is my unique contribution. Does HFSP help in preparing the scientific idea in the applicant of a proper format before final submission? No, sorry. If you need some help, ask your host supervisor. If you're not a native English speaker, get someone who is to read through it for you, because that can be really, really helpful. If you don't want to, well, no, you really should be working with your supervisor. They should be helping. But if you also want an extra opinion, try and find someone who's an expert in the field to give you a quick peer review. That's also really helpful. And all of those things should really help you. Is it allowed to use preliminary data that's unpublished from the host lab in my proposal? Is it good to, or better avoid? It depends on whether you're showing then that this is just an incremental move up from what the host lab have already done, or whether that preliminary data is being used to show that a method is feasible. So again, it's up to you to decide whether it's a good idea or a bad idea to use the preliminary data. We don't say yes or no. It's completely up to you what you want to do.
Maybe I can just add to that. Make sure that if you are including preliminary data from your host lab, that your host supervisor is well aware of that, in case they're not ready for reviewers and external reviewers to see this preliminary work. So just keep that in mind.
Perfect, Elise. Thank you. Are there grant schemes that you can apply for only if you've been an HFSP fellow? No, there used to be a CDA scheme in the past, but there's no longer a CDA scheme. So there is not a sort of closed loop where you can go on to apply for something else. But as we said before, having had funding, it's really useful for then applying for funding in the future. Is the 3-year restriction after PhD completion counted up to the call opening? This is an eligibility one. I'm going to give it to you, Elise, because you're right there.
Yeah, I can quickly answer that. Yes, so it's 3 years before the full proposal submission date. So this year, the full proposal submission date is the 24th of September, 2026. So 3 years before that, the 24th of September. Did I say October or did I say September? Just
you
before?
said September. It's okay.
Great. So if you got your PhD conferral, so your PhD certificate, if that's dated earlier than the 24th of September 2023, you may be ineligible. If you have any post-PhD career interruptions like medical leave or parental leave or caregiver leave, please contact us with all the dates. So the dates of your PhD, the dates of your leave, how much time you've been on leave and things like that. And we can try to get you an exemption, but like I said, please contact us. And if you are right on the cusp, like a few weeks around that date, please also contact us and we will see what we can do. But in general, that is the guidelines there. So if you have your PhD before the 24th of September 2023, you might be ineligible.
Excellent. Thanks, Elise. That's perfect. Is it necessary that my host supervisor is from the life science background? No, but you do want to show that there is enough expertise and understanding in the field that you're moving into to support you. So if you're going to a host supervisor who's got a really strong, let's say maths background, you might want to find a second supervisor who's got expertise in that kind of life science field that you're moving in, that you actually want to move into as well, just to make sure that it's really clear that the expertise and support is there for you. Can we have a paper already published with the host lab, which is different from the PhD project? I think we've answered one very similar to this before, but no, unfortunately you can't. There are some very, very odd exceptions like consortium papers, but please just get in touch with us and ask, because then we can give you a really clear answer. And it's very difficult without seeing what the paper is and understanding the context for us to say yes or no live. Thanks.
Maybe I can also,
oh.
follow on that. Just like the thought in general is that we're looking for the most ambitious people who are going to someone they've never worked with before, going to a new country they've never lived or worked in before. You know, you're changing everything. You're changing your project and who you're going to be working with. So if it's someone that you have done a paper with before, that you've got a publication with, it's quite likely that you're not going to be eligible. Do check with us. Like Carolyn said, if you are just, in a consortium with 40 other people and the other host supervisor, your proposed host supervisor, just so happens to be on that paper, maybe we can talk about it, but please send us an email if you have a publication with your proposed host supervisor and you would like to be considered for an exemption.
Thanks, Elise.
Thanks.
No, it's good. That's a really lovely explanation. I think sometimes we get lost in the details of answering the questions, so that's perfect. I applied in 2024 for an HFSP from India in collaboration with CNRS in France, and I want to ask if it's still accepting the translational project. No, we don't. So as before, the rules are exactly the same. We still don't accept translational projects. I'm really sorry. You definitely want to go to a medical research funder for those. We really are looking for those exciting, novel, frontier, basic biology projects. So not looking for a cure or a drug, not looking to develop a piece of technology, looking to really understand a fundamental biological question. I work with mosquitoes, but I'd like to work with ticks. Is that suitable for an LTF? The answer is you have to make the argument. It's up to you. You have to show why it's exciting, why it's new, why it's different to you, why it's different from the work you've been doing before. So, yeah. Can I apply with my current host supervisor? I've just joined 2 months back and I don't have any publications with my current host supervisor. So it's, again, I would say this is one of those ones where you need to send us an email and we will check, because we always need the really specific dates for this, and it's very difficult for us to just turn around and say yes, no. Send us an email, send us the dates, send us the information, and we can check. How many letters of recommendation do we need to submit, and how recent should our referees be? Carolyn is answering that, so we'll leave it. And then, is cognitive behavioral neuroscience in humans considered life science? Sandra, you can answer this, because the answer is?
I very much hope so.
Yeah. Excellent. Thank you. Okay, so I think we have come to the end of our questions now, which means we're going to come to the end of our session. So just as we're coming to the end, I'd like to say a massive thank you, first of all, to Liliana for hosting and doing all the wonderful work in the background. Second, to Carolyn, who none of you have seen, but she's there actually answering loads and loads of questions and doing everything online. So thank you, Carolyn. A massive thank you to Elise as well, especially for all those excellent videos, but also for answering questions. It was really helpful. And a huge thank you to Sandra for such an excellent presentation, and also being such a wonderful presence and giving people a real idea of what an HFSP fellow is like. And also just finally, a quick thank you to Guntram for such a lovely explanation of the HFSP organization, and then to all of you for joining us. I think that Liliana will just be posting in the chat. There's a quick evaluation that will pop up after the webinar's over. So if you could fill that in for us, that would be great. And thanks so much. Have a lovely morning, evening, afternoon, wherever you are. Thanks, everyone. Goodbye.